that basically predict the delta G value based on the parameters fed into the equation. And what are those parameters? Those parameters are the structural parameters that are derived from the protein RNA complexes. Then every time we take a partial derivative of the equation. Why we need to take a partial derivative? Because we wanted to.
Professor Ranjit Prasad Bahadur, sir, Professor in the Department of Biotechnology, Indian Institute of Technology, Kharagpur, and by Professor Pritish Bharadwaj, sir, Professor in the Department of Applied Sciences, IIIT Allahabad. Professor Ranjit Prasad Bahadur, sir, will deliver his talk on molecular architecture of protein RNA recognition, and Professor Pritish Bharadwaj, sir, will deliver his talk on data and challenges in computer-aided drug designing. As per the AICTE guidelines, the session will be recorded.
Places which are
Form will be shared during the session for the participants to mark their attendance. If participants have any queries during the session, they are requested to write it down in the chat box. Now, Dr. Nidhi.
Places which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein.
On behalf of organizing committee of this sponsored 2-week FDP and computational biology in therapeutics and theranostics. So today we have Dr. Ranjit Prasad Bahadur, who is a professor in the Department of Biotechnology, IIT Kharagpur. He runs computational structural biology lab in the Department of Biotechnology at IIT Kharagpur. Professor
Places which are
Prasad Bahadur obtained his bachelor's degree in physics and master's degree in electronics from the University of Calcutta. Thereafter,
places which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA.
to obtain a PhD in interdisciplinary field of bioinformatics and computational biology. So he has developed many computational tools to study mechanism behind a biology.
Places which are
the main focus of his doctorate thesis was to understand the structural basis of interactions, mainly focusing on protein-protein interactions. So he has devoted his time to study how a protein folds. So protein folding, which is a mechanism that we all would like to know, has been this topic of interest since years. He has
places which are
asked to solve different problems in this field. During his postdoctoral period, he has studied many new systems which assemble to build a capsid in viruses. So, how does a protein specifically recognize the RNA molecule and dynamics behavior of the cellular environment? This has been his topic of interest, and he has well-established, well-published
places which are
reputable journals like Proteins, Journals of Molecular Biology, Journal of Proteome Research, and this area of specific and non-specific protein-protein interactions has been well studied by him. We are fortunate enough to have him with us today so that he will unfold all this mystery of protein RNA recognition before you all. So I happily welcome
places which are
Prasad Bahadur to this FDP, and we acknowledge that he has received, he received our invitation and he agreed to deliver this talk amidst his busy schedule. So we welcome you, Professor Ranjit Prasad Bahadur, and now he will be addressing the audience. So it's over to you, Professor Ranjit Prasad Bahadur.
Places which are hello.
Yes, no problem, sir.
Okay, good. And can you see my slides?
Yes, it's visible, sir. Visible.
Can you see my slides?
Yes, sir, it's visible.
Okay, good. Okay. Before I start, I should thank Dr. Mishra for this nice introduction. and places which are. Thank Dr. Shamanto for his invitation to present my work in this lecture series. Just a minute. I Okay, so and I also like to thank the university or the institution IIIT Allahabad for this nice for arranging this nice program for I think it's a QIP program, right? Places which are Dr. Shamanto.
Yes, sir. Yes, sir. This is a
Okay,
FDP
okay,
program.
okay,
Ah.
okay, good. Okay, good. Thank you. So, can I start now?
Yes, sir. You can.
Okay, good. I think you can see my video, my PowerPoint, right? There is no
Yes.
problem.
No, no, no.
Okay, so as Dr. Mishra already mentioned that I am a computational biologist by training. I did my PhD in computational places which are, and I work mainly in the field of protein RNA interactions, and we are trying to develop some algorithms, databases, and web servers, and we try to understand the molecular architecture, or basically the we do the structural analysis to try to understand the structural basis of these recognition processes. So my lab is situated at the biotechnology department of IIM places which are now, my work, so as I told you that we are mainly interested in understanding the structural basis of protein RNA interactions, and for doing so, we are developing several algorithms, and that includes the development of protein RNA docking benchmark. I will come to that docking benchmark in detail in my talk, later part of my talk. And we also places which are for prediction of binding affinity and binding hotspots, so that I am going to cover these two topics also. And we also develop scoring functions for docking, especially focusing on protein nucleic acid docking, and conformational analysis of RNA binding proteins that interact with RNA. You know that protein RNA interactions are almost every time are driven by conformational change places which are like a rigid structure. They are flexible molecules, so they change their conformation upon association. So what kind of conformation they change, that is very important to decide their partner. And I am not going to cover the intrinsically disordered regions of proteins, because that is another topic, but our lab also work in the area of intrinsic disorder, especially looking at the intrinsic places which are in the RNA binding proteins, as well as to study the role of water molecules in this recognition process. You know that water molecules are everywhere inside the cell, and the major content of this any living organism or any living cells is water. So all the interactions or associations are taking place in the aqueous environment. So water molecules takes a major role in almost all reactions. So we also look into the places which are molecules in these reactions. Other part that we are working, that is mainly on non-coding RNA, and we are not. I'm not going to talk on that part. That is another aspect of our activities in CSB lab. So we develop basically algorithm for prediction of miRNA, LNCRNA, and how these ncRNAs are basically regulating the gene regulation. They are involvement in the gene places which are the comparative genomics of these non-coding RNAs. That those activities also we perform. And the third part, basically, it was my long back interest, but I'm not going to talk anything about that one. So in this today's topic, today's talk, mainly I will focus on the structural analysis of protein RNA interactions, and how we can places which are, or how we can use the information on the structural data that are available in the different web server, and draw some meaningful biological insights to understand the protein RNA recognition. So
Sir,
yeah.
sorry to interfere. Sir, because the slide is not moving. Now it start moving.
Okay,
Okay.
okay.
Uh,
Just a minute, because it was my Can you see the movement of slide?
Yeah.
Places which are.
Move to the third slide. Can you please try once more before and after? Can
So
you repeat once?
this is my main slide. Then this is the first slide. Can you see the movement
Now
of the
the
slide?
slide is on third position. Now it's showing third slide.
Okay, okay. I think
Just
when
do it
I'm showing
slide mode. Sir, do it in slide mode.
now it is okay. Now it is okay. I am changing the
Yeah, now
slide.
it's
Can you
more.
see?
Yes, yes. Now it's okay.
Okay. So when I do the presentation mode, there are places which are okay.
Yes, I
Fine. Now
think you can,
it is
sir,
visible,
you can
right?
do in presentation mode. I think in slide. If
Yeah. Fine. Fine. No problem. Now it is okay, right?
yeah, now it's okay, but you can do it presentation mode.
Means full screen.
Yes, sir. I mean, this is full screen. You can do it.
But when I do full screen, then maybe changing of slide there is a problem.
No, no. Just you can
Now,
try once, sir. You can try once, please.
now I'm trying. Okay. Can you see the changes in slide?
No, one.
Places which are.
It has not come. It's not been full screen, but
Slideshow present online.
no, sir. नीचे भी कर सकते हैं, sir. Is there where is percentage?
Now can you see the changing of slide places which are, or it
Okay,
is stuck to the same slide?
now it's sliding two, sir. Now in second slide.
It's not sliding, right?
It is second slide. You go to the third slide.
I have changed into the third slide, but it is not working. I think you can't see.
No, it's still there in second slide itself.
There is some issues I think with. So I places which are show there is a cup in the bottom, right?
Yes, yes. You try once, sir.
So now I am showing the main slide. Okay, what slide
Yes.
you are seeing?
First slide only, main slide.
Now I changed to the second slide. Can you see?
No, it's remaining the first slide itself.
Okay, that is
Now
the
it's
issue.
coming. It's coming to the second slide. That's a bit slow, I think. Now go to the third one, sir. You go to the third one.
Places which are. Can you see the third slide?
No, still there is second slide. Uh, but in that case,
Or what you can do?
can I in that case?
What I places which are.
Sir, can I just do one thing? Can I just try once you sharing your slide? Maybe from my side, I can do it better. Can I do that?
I cannot hear you very much echoing.
अच्छा ma'am आप please mute yourself ma'am, lot of noise is coming. Nidhi ma'am, please mute yourself. अच्छा
Places which are.
now.
Yeah, it's okay.
अच्छा, one thing sir, can I share your slide?
Yes, you can share my slide.
If please stop sharing, then I can try once maybe.
Okay. Stop share. And maybe this maybe issue with the bandwidth.
Maybe.
Yeah.
So is it visible everyone?
Yeah, I can see you.
So you
I
can see me now.
Yeah, I can see the places which are.
Moving to slide second.
Yes, sir.
Okay, it's working. Yeah.
Third.
Yeah, it's okay.
Yes.
Sir, okay sir, then I'm monitoring. आप just you tell me to move, then I will end. Okay?
Okay.
So I think this slide, I think
So,
you were there.
yeah, this slide. Okay, so I think we all know to the basic concept of central dogma, but still there are some challenges on this central dogma, because we believe that places which are RNA and then we got a protein. But anyway, protein RNA interactions is essential for regulating the gene expression and the expression. There are different kind of RNA molecules are nowadays invented, and they are involved in various types of gene regulatory network. So in this topic, I shall specifically look into the protein RNA complexes. Those are complexes which are and available in the protein data bank. Now, I think many of you or almost all of you heard about protein data bank. So it is hosted in www.rcsb.org. So just go to the next slide. Change the slide, please. Hello. Hello. Can you change the slide? Places which are. Can you hear me? I can't hear anybody.
Sir, I have changed.
Okay, so it is a lag in maybe here in my system. Okay,
My.
okay, okay, fine. So, but it's places which are in full screen mode or
No.
I means the slide is not in full screen mode. Now it's fine. Yeah, yes. Okay, so if you look into the PDB, the protein data bank, which is nothing but the three-dimensional structural complexes which are bound with the ribosomal RNA, then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form and 100,000 structures, and of these, only around 5% are the complexes with protein nucleic acids. So why the protein nucleic acid structures are very low? Because one of the main reason is that the co-crystallization with protein and RNA, especially RNA, DNA is not that much a problem, but co-crystallization with RNA with protein is very difficult because of the flexibility of the RNA structures. Places which are structures of RNA is quite flexible. You know that RNA can have different kind of structures, not like the DNA. Most of the DNA we are having B form, nice Watson-Crick double complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form, and the amount of data to study the interactions or to understand the structural different kind of interactions, what are the different kind of forces that basically drive the interactions and the stability of the complexes. Okay, go to the next slide, please. Complexes which are okay. So now when we talk about the mentioned you that protein RNA interactions are very much flexible. They are not like a rigid body interactions. They are flexible kind of flexibility. You need to know the conformations of the both protein and RNA complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that the bound state and one is the unbound state. So we must have the structures, both bound and unbound states of the corresponding protein and RNA molecule to understand how the conformational changes govern the complex formation. So that is one of the biggest issue or challenges to study the conformational changes that drives the protein RNA recognition. And these conformation places which are often responsible for generating the specificity and affinity of the recognition processes. We all know that mutations occurs in proteins or especially related to some diseases. So these mutagenesis studies also reveal that the substitution of functional group at the recognition site places which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means the binding may not happen. So mutation is a very much important from the perspective of the disease biology, and we find that many diseases are associated, and these mutations causes different kind of change in the native interactions which eventually leads to the perturbation in the complex formation. And complexes which are regions in the RB protein, because you know that initially when structural biology started, or people thought that the evolution or the evolution was studied in terms of the sequence. Like you know that blast was complexes which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that to study the sequence-based similarity. But later on, when the various structures of the homologous complexes came out, people understand that it is much more important in terms of evolution to study the structural structures of the molecules rather than the sequence. So the best example I always cite is the myoglobin structure and the hemoglobin structure. Both of them have remarkable same structural complexes which are compared to the sequences between the myoglobin is a monomer, that is it has only one protein chain. Hemoglobin is a tetramer or a dimer of two dimers, alpha and beta chains. But if you compare at the sequence level between the alpha chain of hemoglobin or the beta chain of hemoglobin with the single chain or monomeric chain of myoglobin, you find less than 20% complexes which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means the structural fold, they are remarkably similar, and both of them basically almost do the same function. So you see that, so if you want to draw the conclusion based on their sequence similarity, then only based on the sequence similarity, then you might have not do justice. So you need a structural information. That means their binding site, the scaffold that E and F, H complexes which are bind to the porphyrin ring of the hemoglobin and myoglobin, their fold is basically a globin fold, and that is preserved during the evolution. So so that's why it is very important to understand the conserved region in the macromolecular structures, especially at the binding sites. And these evolutionary conserved residues can complexes which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that because if you mutate this one of these evolutionary conserved residues, then the binding affinity can drastically change, and it can have very strong consequences on the binding. Yes, somebody wanted to say something. Okay. Can you go to the next slide, please? Complexes which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it the complexes with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein RNA complex, a protein binds to the single-stranded RNA, we call it a single-stranded complexes with single-stranded RNA. Next slide. Next slide, please. Okay, so first I'll talk about the development of the protein RNA docking benchmark. Why we need a docking benchmark? Because we know that a crystallization process to determine the 3D structures of macromolecules is very tedious and time consuming, laborious, as well as very expensive. So alternatively, people or scientists have thought to develop some in silico methods, complexes which are bound with a ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it the complexes with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein RNA complex, a protein binds to the single-stranded RNA, we call it a single-stranded complexes with single-stranded RNA. Next slide. In the binding partner. So that's why docking benchmark is nothing but a set of protein RNA complexes, gold standard set of protein RNA complexes, which can be used to test the developed algorithm. What kind of developed algorithm? That means the scientists who are developing the algorithm for predicting the protein RNA complexes on given protein and RNA structures. So this algorithm is basically takes the protein and RNA structures, 3D complexes which are in the unbound complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form, and if a protein RNA complex. So we have used the information of protein RNA complexes that are given in the protein RCSPB. So we curate all the protein RNA complexes that are available in the protein data bank. Then we extracted the protein chain and RNA chain from the complexes, and we did the blast search. So these basically complex data sets are basically the bound form. We call it the bound complexes. Okay. Now we have to find complexes which are corresponding unbound protein and unbound RNA, 3D structures of unbound protein and unbound RNA in the PDB, so that we can have complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein RNA complex, we extract the protein chain and RNA chain from the complex, and then we do the blast search to find out their unbound partner. So when we find the exact very high identical sequences in the unbound form, we take the protein and RNA as their unbound form. And this way and for doing this process, we just did some check here. So resolution of the X-ray structures or the complexes, we should have less than 3 angstrom resolution. Complexes which are the complexes that we are select, basically more than 30 amino acid residues, and RNA should contain more than 5 nucleotides. Then we check the sequence complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein RNA complex of 35%, that means none of the two sequences are less than are more than 35% identical in the data set. Then we find out their secondary structures, especially based on the different type of complexes, and we divided these complexes into 4 structural subgroups. One we call it a tRNA, that means the complexes having tRNA. Then we have ribosomal RNA, that means complexes which are bound with the ribosomal RNA. Then we have a single-stranded RNA and duplex RNA. That means that sometimes you know that RNA can form a double strand. That's why we call it a complex with duplex RNA. And most of the cases, RNA found in the single-strand form. And if a protein RNA complex, a protein binds with a single-stranded RNA, we call it a single-stranded complexes with single-stranded RNA. Next slide. Okay, so we found around 126 non-redundant. So you should remember this is a non-redundant set of protein structures, 126 protein RNA complexes, of which we can found in 21 cases out of 126. For 21 complexes, we have both unbound protein and unbound RNA. In 95 cases, we have only the unbound protein, not the unbound RNA, because I told you that crystallization of single unbound RNA is very difficult due to its flexibility. And in bound unbound case means the protein is in the bound form, but the RNA is in the unbound form. So that those are only 10 cases we have found such complexes. So and you can find this docking RNA benchmark dataset in this website. Next slide, please. Okay. Now, as I told you that when protein RNA recognition takes place, so there is a conformational changes either in the protein side or in the RNA side, or it can have affect the both protein and RNA. So how you can check the conformational changes? So there are few parameters looking at which you can identify the change in the conformation. One is the C alpha RMSD. That means you calculate the you superimpose the protein structure in the bound and unbound stage. A particular protein, say X1, you superpose the 3D structure of X1 in the unbound state and in the bound state, and then you see the C alpha RMSD, you calculate the C alpha RMSD, and then looking at the C alpha RMSD, you can quantify the change in backbone conformation. And what is PRMSD? You do the same thing for the RNA side, and you take in that case the phosphorus atom in the RNA to check the backbone RMSD of the RNA. And if you combine both C alpha and P, you get the IRMSD is nothing but the RMSD of the interface residues, because you know that any interactions, there are some residues which are at the interface between the two interacting molecules. There are many residues which do not participate in the interaction. Those are especially required for other purposes, like many if we talk about protein interactions, you know that when protein folds, there are many residues which part of the core of the protein, which basically involved in the folding of the process. There are surface residues which are basically involved with the interactions with the surrounding to get or to give the hydrophilic interactions with the surrounding. And there are part of the residues which are responsible for the interacts with their partner molecule. So those are called the interface residues, or and if you calculate the RMSD of those residues, that is called the IRMSD. So why we are showing here IRMSD, CRMSD, and PRMSD? Because when a protein interacts with RNA, so it may so happen that its interface residues can have different kind of conformational changes compared to those residues which are not part of the interface. That means which are not interacting with the RNA. So that is called the other residues, or the you can also calculate those residues to have a look, or to have a check that whether those residues are having any change in the conformation when it binds. So that's why you segregate it into IRMSD and CRMSD. IRMSD is nothing but the RMSD involving the interacting residues. CRMSD is the RMSD which are not interacting um, with the RNA molecules, and PRMSD is the RMSD with the for the RNA backbone, that is for phosphorus. Now, if you look into the values, you can find that many complexes, their conformational changes are quite less than 1.5 angstrom, and in such cases we call it a rigid body association. That means there is not much conformational changes upon binding with the RNA and protein. Then we can say semi-flexible complexes, where the IRMSD changes between 1.5 angstrom to 3 angstrom, and the full flexible complexes, or the interaction is fully flexible, where the IRMSD is beyond 3 angstrom. So these are the number of different cases, and that are represented in our docking benchmark data set. Now, when you look into the hello. Okay. Now, when you look into the PRMSD and CRMSD, yes, any question?
No issue.
Okay. So if you look into, as I told you many times, that RNA molecules is much more flexible than the corresponding protein. So if you look into the C alpha RMSD or PRMSD, in the almost all the cases, you find that PRMSD is much higher than the C alpha RMSD. That means RNA undergoes large conformational changes than its partner protein, and these conformational changes can often induce the binding process. Okay, so that is called the induced binding. So basically, there are two modes of binding phenomena. One we call lock and key model, which is a basically rigid body association happens in the lock and key. So the binding sites are the preformed like a lock and the key. So there not much conformational changes are observed in those cases when this lock and key model are present. And another is called induced fit. That means binding partner changes their conformation based on their size or shape of the partner molecule. So that is called the induced fit. So in protein RNA conformation, often we can see that this induced fit are occur in both either of the protein side or RNA side, or sometimes in the both the binding partners. Okay, next slide please. Okay, so these are few examples. The first one is example of the full flexible. Here you can see the green is green. Actually, here the superimposed structures of the unbound protein and unbound RNA, unbound protein along with the unbound RNA and bound RNA. you see that in the first case, the green one is the bound protein structure, and cyan one is the unbound protein structure. You see the complete domain movement is taken place upon binding with the tRNA. So this is a significant change in the conformation. That's why we call it a fully flexible recognition or the association. In the second complex, you see the little bit of movement of the some regions, or if you see some loop region here changes its conformation, say green and cyan. So here there is not much, no such domain movement is offered, but there are some change in the conformation in the secondary structures or in the loop region, which makes it a flexible recognition. And if you look into the figure C, it's basically a sorry, this is a rigid body association, the B1, because not much conformational changes is found here. And if you look into the C here, it is basically a semi-flexible association, and you can see that there are a little bit of changes here. You can see the cyan and this green is not matching, superposed. That means there is a change in the conformation upon complex formation. So this way you can grossly divide. You cannot say so you can grossly analyze the structures or analyze the conformational changes into these three main categories. Next slide, please. Next slide, please.
Has not been changed?
It's changing, sir.
It's changing now.
Yes, sir.
1 second. I think to him it's not visible. Sir, can you see? It's hello? Sir, it's changing. I think he got disconnected or what? Hello? Hello? Excuse me, am I audible?
Yes, sir. You are.
Yes, yes, we can hear you. I think Professor has lost his contact.
I think he got disconnected. Let me call him. So have you joined?
No.
Please, audience, sorry for the inconvenience. I have called him. He will be joining soon.
Yes, he is joined.
Okay, sir, have you joined? Hello, can you hear me, sir?
Can you hear me?
Yes, yes,
Okay,
yes, yes.
I think it's a problem.
Sir,
Internet issues.
is
Okay.
the slide visible now?
Yes, I can see.
Okay. So basically you are here. Okay.
No, no, I think no.
Yes, sir. You have done
Yes.
over
Okay.
here.
That.
What
Okay.
other
Okay.
specific
Yes.
requirement?
Okay. Fine. Yes. Yes. This is done. And then next we are going to discuss the development of a mathematical model for accurate prediction of protein RNA binding affinity using structural parameter. So in wet lab, there are different biophysical methods to predict the binding affinity or to calculate the delta G, or from KD also you can calculate the delta G value or delta G value. It's a complex association. People also try to develop some computational algorithm so that this algorithm can use the some structural parameter information of some structural parameter and then predict the binding affinity. So that is what how we are going to see how we can predict the binding affinity using some structural parameter of macromolecular complexes. Next slide. Yes. So similarly, so again, so for calculating the binding affinity, it's interactions between the two molecules. In this case, it's a protein and RNA. So we need the both bound and unbound structure of the complexes. And then we first curated a data set which contains the KD value, specifically reported KD value in the literature. So for all the protein RNA complexes that are present in the docking benchmark, we curate the literature from the PubMed, and then we find the value of the corresponding complexes, and then we calculate the delta G value from this KD using that famous equation ln equals to minus RTL and KD, and then we make a data set of protein RNA complexes with binding affinity. Next, please. Okay, so here you can see that, so these are basically the observed data set or the curated data set, and how the binding affinity is varying. So the average delta G is we found it is highest for the complexes that are found in the single involving with the single stranded RNA, and lowest for the complexes with duplex RNA. Next, please. Okay. Now, this for these complexes, we also found the mutation data set. There is a database, mutational database. If you look into the mutational database, there are many structures that are reported. So the effect of changing delta G due to the mutation. So in the previous slide, what I showed you, those are basically the delta G value for the wild type complexes. And for each of those complexes, we also look into these mutational databases, and we try to find whether there are the reported value of delta G due to the mutation, so that we can compare the effect of mutation on the on this binding processes. So in such cases, we found 123 mutations out of 15 complexes for which the KD values please. Next slide. Along with different parameters. So I will show you what are the structural parameters that we have used. We have the information of the delta G that I have shown you 2 slides before, the curated for each and every complexes from the literature. Then we have generated set of numerical equations to predict the delta G value using those parameters. So it's a set of equations. It's a computationally generated equations that that basically predict the delta G value based on the parameters fed into the equation. And what are those parameters? Those parameters are the structural parameters that are derived from the protein RNA complexes. Then every time we take a partial derivative of the equation. Why we need to take a partial derivative? Because we wanted to change. Derivative is nothing but the change of some phenomena with respect to time or with respect to some other parameter. One is called the independent parameter and one is called the dependent parameter. So change in delta G, let us say, with respect to change in some parameter in our case. Why we wanted to study that? Because we wanted to see whether the changing in the structural parameter can give us a much more accurate value of the delta G. So this way we do every time, and we try to go to the as close as to the experimentally generated delta G value. So this way we optimize the process, and when we find that that our predicted value is very close, or the standard error is very low, so we select that model as a perfect model for prediction of the delta G value. So this method is basically was very nicely reported in one of the science paper by Smith and Lipson, how you can predict the accurate or how you can build a mathematical model based on the some observed phenomena. It can you can apply this basically they have applied this model in some prediction of some geological phenomena. So you can apply this model to any of social prediction or some scientific prediction model you can develop if you have some data set or if you have some parameters to describe the system. Okay, next. Okay, so then what are the parameters that basically used in this prediction model? So definitely the amino acid number of amino acid residues that are present at the interface, number of nucleotides present at the interface water molecules, because I told you the water molecules plays an important role in the binding process. Then a number of conserved or preserved interface water molecules, that means the water molecules that are preserved during the association process. Number of water bridges, that means many water molecules make hydrogen bonds both with the protein and RNA side. Those are called the bridging water molecules. That means it partner. Then there are some quantified parameters that basically tells you the dry and wet interface, the hydrogen bonds, then the contribution of the non-polar area in the binding process, then contribution of the buried atoms in the binding region, then the density of the protein and RNA density of the protein and RNA atoms at the interface, then the number of salt bridges, how many salt bridge interactions are there, stacking interactions. You know that protein RNA interactions are often governed by the stacking interactions between the nucleotide or the RNA bases along with the side chain of the aromatic amino acid residues, because aromatic amino acid residues has a circular or a planar side chain, and this plane can stack with the side chain of the or stack with the bases, and it can make a stacking interactions. and then shape complementary indexes, because as I told you, the shape is a very important phenomena for the recognition processes, whether it is a enzyme substrate interactions or it's a other kind of interaction. So often macromolecules, they try to look into the complementarity shape so that it can bind very tightly with its complementarity shape. Like lock and key model is one of such examples of shape complementarity. And then there is a gap volume index. It's basically calculated the how the gaps are present in the interface, because protein and RNA molecules or any macromolecules, their surfaces are not flat. So if you have a two flat surfaces, the gaps are almost zero. So there should not be any gaps when you join the two flat surfaces. Like you can imagine two pages of a paper. You are taking two papers and making a connection, or you can merge the two papers. There should not be any gap. But protein surfaces are not like a planar surfaces. They are having a clefts and crevices in the protein surfaces or RNA surfaces. So there must be a gap in the surface interacting region. So this gap also plays an important role when the two molecules recognize each other. So these are the different parameters that we have used to define the or to define the equation. Next. Okay. Then we fit into that model, mathematical model. We find some equations here with some of the parameters. Like you can see that FNP is the contribution of the nonpolar area, and we have the dr that define the wet and dry interface, along with some change in conformation, or that C alpha or p value that gives you the better model for prediction of the delta G value. Next. Okay, so this is the our prediction model is basically on the x-axis, and experimental data I think along the y-axis. No, sorry, it's, experimental I think in the x-axis, and predict is along the y-axis. Anyway, the you can see the R square value is 0.92. That means it's a quite efficient method, and absolute error is quite low, 0.27. Okay, next. So we have validated this model with some mutational databases. So the mutation database is already available for this particular protein 1ASY. It's nothing but aspartyl tRNA synthetase. So if you look into this paper by Ariani and Gangloff, so they have done a series of mutations on this tRNA by tRNA synthetases, and they report the delta G value. So what we did basically, we generate all the structures corresponding to these mutations. And for all the structures, we calculate the delta G value from the structural parameter. And then we predict the delta G value to see whether they are closely predicted with the experimentally delta G value. So we found, you see that our standard error is quite low, only 1.69, and mean absolute error is 1.42, and relative error is 16%. So its prediction is quite good, around 90% accuracy we can get. Next, please. So this way we can validate our prediction model, and we can say that okay, our mathematical model is quite efficient to predict the binding effect. Next is the binding hotspots. As I told you in my starting slides, that evolution or conservation during the evolution, or conservation of the structurally conserved region, or the binding site region, or the active site region plays an important role in the recognition process. And if you look into the enzyme substrate interactions, most of the enzyme sites are evolutionary conserved. Serine thionein kinases, they are evolutionary conserved. That motif is evolutionary conserved. Whatever the changes in the other part of the protein might take place during the evolution, but the enzyme, the active site remain conserved during the evolution. So that observation of that phenomena we have utilized here to predict the binding hotspot at the protein RNA interfaces. As I defined you, the hotspots are those mutations which can drastically change the binding affinity of the complex or the interactions, and it can have a very crucial effect in the recognition process and the cell functioning. Next slide, please. Okay, so here what we have used the HSSP database. So these are basically a homology derived secondary structure of protein. That means in nutshell, it is basically gives you the 3D superposed structures of all the complexes or all the protein structures that are available in the PDB. Like BLAST, what gives you BLAST basically gives the alignment based on the sequence. So it gives based on the alphabet. It doesn't do the structural superposition. It's just read the sequences as a string, as an alphabet. So it's a sequence based method. But HSSP it does basically the structure based method. It does the structural superposition, and then it gives the alignment based on the structural superposition. So as I told you that when you do the evolutionary study, it is much more important to address the structural superposition or structural conservation rather than the sequence information. That's why we took the HSSP database. Next. Okay, so this is just a model for how we can identify the interior interface and surface of a protein. So left hand side you can see it's a complex protein. You can see one is protein, another is RNA. So this is the interior of the protein that you can see. It's a core of the protein that basically gives the stability of the tertiary structure of the protein. This is the interface region, and this is the surface region. So you can calculate the solvent accessible surface area of this complex to calculate how much surface is buried upon complexation. Next, please. And we can use Shannon entropy. So it's nothing but gives you a sequence variability of a given position, which is given by this equation. So SI is the ith position, the sequence Shannon entropy or the sequence variability that can be given by this equation pk ln pk. I am explaining this how you can get this value. Go to the next slide. Let us say you have a query sequence like which is shown in a VTISC TGS. So let us say your sequence is 8 residue long, and when you do the structure based alignment, you get 10 other sequences or 10 other structures which are aligned to this query sequence. Now it has 10 position 1 2 3 4 5 6 7 8. Your let us say your protein is 8 residue long, not 10, and you have 10 aligned other protein structures which are aligned to this sequence. Now how to calculate the entropy of the first position? So you can look into the first position is V, and in out of 10 how many positions V is there? V is 5 positions. 1 2 3 4 5 V is there. So what is the probability of V appearing in the 5 position? Is 5 out of 10 ln 5 out of 10. Then you look into the what are the other variables. You have 2 gaps. So 2 to probability of appearing 2 gaps out of 10 is 2 by 10 ln 2 by 10. What are the other option is there? You have alanine. So how many alanine are there? You have 3 alanine. So probability of appearing alanine at position 1 is 3 by 10 out of 3, ln 3 by 10. So this way you calculate the entropy of first position. Then you calculate the entropy of the second position in the same manner. So entropy of the second position, you can see you have 1 2 3 4 5 t. So probability of t is 5 by 10 ln 5 by 10. Then you have r, only one r, then 1 by 10 ln 1 by 10. Then any problem? You cannot see. Is it a problem with my sound?
See and hear. I think the problem is on her side.
No. It's fine. No issue.
Am I audible to others?
Yes, sir. You are audible.
Okay, fine. Okay. So what I am telling, if you want to calculate the entropy of the entropy is nothing but sequence variability. Means we are assuming that these 10 sequences are from the different species during the evolution, and you see first position how this position is changing. In some sequence you find V valine, some sequence may be deleted due to the evolution. In some sequences you can see alanine is there instead of valine. So this way, how this first position is changing during the evolution, you are basically quantifying the position with this mathematical equation. Second position also you can same way you can calculate the variability of the second position. This way you can calculate all the 8 position of your sequence and see how the position is changing during the evolution. You know that not all the residues are changing during evolution, right? When you do a blast search or when you do the cluster W alignment with all your blaster sequence aligned sequences, you see some portions are green, means you see a stretch of portions which are identical. Some of them are similarity, 1 star, 2 stars, 3 star. Identical means same residues are present in that particular position. Similarity means you have a similar kind of substitution, like in one sequence you have valine, in another sequence you might have isoleucine, because both of them are hydrophobic amino acid residues in the same position. But you cannot quantify. Quantitatively you cannot say whether the first position is more than the other position. But if you have some mathematical data, obviously looking at this S1, S2, S3, S8 values, you can say which position is evolving faster and which position is evolving slower. Faster means it will have a frequent changes. Now when you have a very frequent changes, your S1 S value, because it is just a probability value, it's a log of probability basically. So your S position S value will be very high if it is very fast. And if it not varies at all, like in our case you have a 10 positions, so in all the position is valuing there, then what value you have? 10 by 10 means 1, ln 10 by 10 means 1, means 0. S1 value would have been 0. That means looking at the S1 value, you can say 0 means this position is fully conserved. That means during evolution it has not evolved at all, it is fully conserved. So this way, looking at the value, you don't have to look manually, because when you do large scale genome wide evolutionary analysis, you cannot look into manually, right? So you need some data, you need some parameter, you need some quantifying parameter. Looking at this parameter, you can easily say that okay, this position is fully conserved because S1 value is 0. That means all the position is having the same residue, it didn't evolve. So this way you can quantitatively say which how a protein sequence based on the structural alignment is evolving. Okay, next. So we have used this phenomena or this Shannon entropy and for our prediction or building the model. So that part is explained, I think, already. Yes. And then we did the analysis for different type of complexes. We found that some of the complexes are evolving faster, some of the complexes are evolving slower, looking at this S parameter. Next, please. This is also, you can, as I told you, you can divide the protein into based on the interior, interface and surface, and we can see that interior is much more conserved based on this parameter compared to the interface and the surface. That means surface residues are evolving faster than the interface residues, and interior residues are basically evolutionary conserved, because these are the core residues of the protein that gives the stability of the 3D structures of the macromolecule. Next, please. Okay, so these are some of the map how the protein evolved. You see that so red region is basically a fully conserved, and blue region is fully evolving, means evolving faster. So this green basically are the RNA molecule. These are the few examples. You see that the binding site, which is basically binds with the RNA, the protein part which is bind with the RNA is mainly red. That means the residues that they are basically evolutionary conserved compared to the other region of the protein, mainly the surface region, which is evolving faster. Next. These are some residue-wise evolutionary phenomena. You can see some of the residues are evolving faster, some of the residues are evolving slower. Again, this is quantified based on the mathematical formula that I explained. Next. Again, we did some mathematical analysis where we have shown that the interior of the protein, that means the core of the protein, is evolutionary conserved than the interface, than the surface. That means on the other way, surface is evolving faster than the interface, than the interior. Next, please. This is another interesting feature that we have found. Some of the residues, when we look into the protein RNA complexes, so many of the binding protein have more than one subunit. That means multimer, means dimer. So in such cases, some of the residues interacting both simultaneously participate in protein-protein interface and protein-RNA interface. So we have found that these residues which are participating both protein-protein and protein-RNA interfaces, they are evolutionary more conserved than the other residues which are participating either with protein-protein interactions or with protein-RNA interactions. Next, please. We also look into the conservation of the water molecules at this size, and we see that the residues which are basically conserved with the water molecule, or the interactions which are conserved upon association, those are evolutionary better conserved than the other residues which are not conserved during the evolution. Next, please. Okay, again we did some analysis with major group and minor group, and we found that the residues which are interacting with the minor group are better conserved than the residues which are interacting with the major group, because you have a completely a different situation when you compare with the DNA major group and minor group is, the physical characteristics features is completely different than the RNA major group and minor group. Next. Okay, so we finally what we did, as I told you, we have the delta G values curated from the literature or the experimental data of 1ASY, and we compare our predicted delta G value with along with this parameter, with the conservation of water, and the conservation of the residues at the binding interfaces. Next. And yeah, these are the parameters that I think already we have discussed: Hall bridges, hydrogen bonds, change in stacking interactions, that have been used for building the model for prediction the binding hotspots. Next. So this is we use random forest method to predict the binding hotspots. So this is the predicted value, and these are the actual delta G. We can see that some of the mispredictions are there, but it's okay because it's a model, and we never say that model could predict 100% accurately. But it is much. It is, I think our prediction is more than around 86 or 87%, and that is quite efficient model for predicting the change in delta G in for a given mutation. Okay, so we have developed a web server that is hosted in this website for if you submit a protein, and it can predict the change in delta G value, and it's called HotSpring, hotspot at protein RNA interfaces. Next. Okay, I think that slide is not important. Means how this data. So this is the output of that web server. Next. Okay, fine. So what we have done, our contribution from the lab in this field. So we have developed a protein RNA docking benchmark, which is basically an essential tool for assessing the docking models. Then we developed a regression-based model for prediction of the binding affinity of protein RNA complexes. And finally, we have developed a web server that basically can efficiently predict the hotspot regions and a given protein RNA interactions. Okay, I think I am almost done. Okay, thank you for your patience. Okay, thank you. I'm done, Dr. Shamanto.
Okay, sir.
If you have any questions or queries. Hello. Can you hear me?
Yes, you are, sir.
Yeah.
So audience, please, if you have any queries, you please write in the chat box or please say.
Hello, good evening. Am I
Hi.
audible?
Yes, audible.
Yeah, sir, I had a question for you.
Yes.
The entire interaction what you mentioned about the protein and the ligands as well as the protein RNA interaction, which is done in an computed atmosphere, right? At the cell, you will
So
find that there are lots of
so when you have a structure, so if you can imagine any structure, when the atomic coordinate of atoms are known, you can calculate the interactions between them. Because atomic structure, you get the coordinate each and every atom, three-dimensional coordinate that are present in your protein and RNA, and using these atoms you can calculate all kinds of possible interactions.
right. Sir, true, sir. But the molecular interactions, if you look at, if you take in molecular biology of consideration, you will see that there is post-transcription translation regulation also. And the
Yes.
amount of protein which is synthesized is always regulated inside the cell environment. That depends on the number of ions which are present inside the cell, the
Yes,
pH
yes,
of the
yes.
cell,
That is
then all the molecules.
one of the yes, yes. So these analyses are entirely done on the X-ray structure, and the hypothesis
Yeah.
and X-ray structures are static in nature, but the hypothesis of the X-ray study or the structural biology is the X-ray structure means it's a crystal structure, and the crystal structure gives you the native conformation. But this is a static information. You cannot generate any dynamic information. If you would like to understand how these interactions are at the time scale, at milliseconds time scale or microseconds time scale, then you have to do some molecular dynamic simulations at the millisecond, nanosecond or microsecond time scale. Then you can have the simulated whatever going on inside the cell, you can have a picture of them, because these are not the
Well,
static molecule, these are the dynamic one.
well,
But what X-ray gives
so,
us, it gives a static picture.
yeah. So, well, you're trying to say is that we can create a model and then we need to test it outside, right?
Yes, you can do the simulation if you want to study the dynamics in detail.
Just to try to understand the chemical kinetics of the interactions that are going, right?
Yes,
Yeah. Okay. But then anyway, the experimental evidence has to be proved by confirming it in the study.
yes, always it is better to have the experimental validation of your prediction model.
Yeah. Do you think that there will be any anomalies there because the model which you have
There is
generated, you
any
might generate and model. Will there be any anomalies based on what I mentioned? Because you might find a model which is fitting in simulations. But when you go for an experimental evidence of it, you will see that there are some anomalies and you don't
yeah,
get that interaction
as
right.
I told you that there is always a scope to improve the experimental methods, because experimental methods are basically based on the available information. If your available information are quite correct or much correct, then your prediction model would be more correct. If your experimental information is like you are getting some information, say you are doing some population study. If your
Yeah.
population study, if you can curate correct information from the say malnutrition in say in India, in some states, if you get a correct information from the data, then the model whatever you build, it will be more correct. I won't say it's 100% accurate, but it will be more correct than if you have a wrong data in your data set. Because
True.
all the prediction, whatever the prediction you are having, like weather forecast, prediction XYZ thing, all are based on observation. Last, like weather forecast, we are doing a particular region, last 100 years data, 150, whatever data we are able with. Or nowadays sometimes what we are doing, we are analyzing the satellite images. So whatever the data we are getting for the prediction model, the model will be accurate whenever you have a accurate data,
Right.
because mathematics cannot tell wrong. What go wrong is
Right.
that the feeded data, if you have noises in the data, then your mathematical model will be wrong.
Right. Sir, let's assume that you get this mathematical calculation correct, and that reciprocates with the experimental studies also. But sir, is there any
Yes,
possibility? Sir, I'm trying to understand. Is there any possibility that the age of that particular individual where you're trying to find out the interaction, that also plays in?
sorry,
Like you have a younger
what is your
individual who
last
is
part?
the
Age of the
age of the
interaction.
yeah,
The
age in the sense the age
age
of the
of the
cell life.
interaction, no, we cannot study age of. So you are telling the half life of these complexes, yeah,
Yeah,
that
true.
we cannot predict from this model the age of the interactions or the half life of the molecule.
Thank you very much. Thank you.
Right. Any other question, please? Okay, then I think you are having next talk ready.
Yes, sir. Yes.
I'm
So,
sorry for 5 minutes late.
I
Okay.
I have just small queries, sir. So
Yes.
generally, when we study the protein DNA interaction, generally we used to do cheap seq. So generally for RNA protein के लिए, what sort of generally test people used to do actually in the lab?
RNA-seq data are there.
That RNA-seq data from RNA-seq data
Mm-hmm.
only they used to get. Okay.
You can do a small RNA library preparation, sequencing, all this stuff you can analyze, right?
So, sir,
Lot of information are there.
so, sir, generally, do you detect any sort of pathogenesis?
No,
here?
no. This, as I told you, this is purely done on the 3D structure.
3D
Those
structure.
chip-seq data or DNA-seq data that don't give you the atomic coordinate of macromolecules. You can get the coordinate only by some spectroscopic tool, either NMR method or X-ray. That you can say that okay, these are the interacting. Okay, fine. Like is2 hybrid protein affinity chromatography that tells you okay, this is the interaction network. That means this protein is interacting with Y, Z, and but you don't know what is their interaction mode. You don't know the 3D coordinate of the interactions.
Correct.
That's for that you need to go for the either X-ray or NMR or cryo, right? To get the atomic structure. If you don't have the atomic structure, you cannot calculate the interactions. Once you have the atomic position of each and every atom, then you can calculate whatever the possible interactions are going on. Because you can have only bonded and non-bonded interactions, right? So you can calculate what are the bonded and non-bonded interactions. It is already known from the physics world. Then you can apply those equations and you can calculate, because you know the coordinate x, y, z coordinate of each and every atom. You can calculate the interactions. Like if you want to calculate the electrostatic interactions, you know the positively charged group, the position of the positively charged group, position of the negatively charged group. You can also calculate the distance because you know the x, y, z coordinate of these two charge groups. Then you can simply apply the Coulomb's law. You can get the Coulomb interactions or electrostatic interaction, right?
Yes, yes, yes. Got it, sir. Got it.
But you cannot calculate those interactions using from chip-seq data because they don't have the coordinate information.
From chip-seq, we can just validate whether they're interacting or not.
Yes. That's you can validate. Yes.
Thank you, sir. It's very nice talk, sir.
Yes, well, welcome, Dr. Shamanto. I also thank you for inviting me for
Thank
this
you, sir,
excellent
for
program.
giving your time.
No, no. I was little bit late. You know that we are going to having another
But anyway, at last we
program,
make out.
two programs, I think. One is that is GN Ramachandran lecture series on 18th, and again we are having BestCon on from 4th to 6th. So we are really means on our toes.
Yes, sir.
You know BestCon, we are organizing BestCon this year.
I don't know, sir.
Okay. You can. Okay, BestCon is organizing by our department this year. So we are very much busy. Sorry for sending the slide in the last moment.
No, sir. Absolutely fine. It's absolutely fine, sir. Thank you so much, sir.
Okay. Thank you.
Okay.
Okay. Can I leave?
Yeah, Nidhi ma'am, are you there?
Yes. Sir, we would like to thank you on behalf of the organizing committee for taking out your precious time and delivering this very informative talk. As a part of the program, we would like to present to you a virtual memento as a token of appreciation from our side, and we will be sharing the certificates from AICTE as soon as we get it. Thank you so much for giving your precious time.
Thank you very much. Okay, thank you very much. Thank you. Okay.
So, participants, we move to our next talk. I think our speaker has already joined. Yes?
I'm here already, ma'am. Thank you.
Yeah. Okay, sir. So our next talk is on data and challenges in computer-aided drug designing. So next speaker is our own faculty member, our own colleague, Professor Pritish Varadwaj from Applied Sciences Department. Professor Pritish Varadwaj is a professor in our department as well as holds the position of dean. He has to his credit more than 150 research papers, several conference proceedings. He has completed several projects. He has guided several PhD students. Before obtaining this position of dean, he has been actively involved in the administrative positions of the institute. He is actively involved in setting up a center of bioinformatics in our department, and if we say the credit of developing this applied science department and bioinformatics course goes to him. So these research areas include computer-aided drug designing, chemical informatics, systems biology, and he would be presenting in you the topic data and challenges in computer-aided drug designing. So we welcome you, Dr. Pratish Kumar Bharadwaj, to take out this precious time because he is a very busy person. So taking out time for delivering this talk, we thank you on behalf of organizing committee. So now he will be presenting his talk, and I welcome him to this.
Am I audible? Am I
Yes,
audible?
even the slide is visible.
Okay, okay. So thank you so much, ma'am. Thank you. I think someone, yeah, please mute on your mic. So there is echo going on. So thank you so much, madam, for this nice introduction. And thank you both of you, like Dr. Sintu and Dr. Nidhi, for giving me this opportunity to address this August gathering. So the entire purpose of this 50 minutes or 45 minutes talk will be just to give you a brief idea that how what are the different challenges right now bioinformatician of the next generation is facing, and will be coming up into some more and more into data, and then what are the challenges associated with this data in the generation of data, as well as like what exactly we supposed to do with this data. I mean, how it is going to solve this purpose in a more refined or better way to come up to a inference system or conclusion. And I hope that if you have any like by end of this session, it is this 1 hour session, if you can appreciate the beauty of the word, more the data better it is, then I think the entire idea of this few slides will be taken into account. So uh, here this the today's talk is we'll be going into more into what different type of data are there in CAD, and then how the things are going in an enormously high speed. And we have all this four paradigm of big data. That means the volume, veracity, velocity, and then I mean we'll see that how the data will make this difference into different inference system within the CAD or computer data designing. So just a minute. So I'll just in one slide we have tried to get into a way that what are the different steps which is currently being within the paradigm of CAD. And in fact, when we started the entire thing around 2002, the CAD was more into three things: QSR, docking, and molecular dynamics. But now we have, with the advance of higher processing speed and the cheap data storage devices, so we have created a lot of different tools or methods or protocols by which we can almost substitute every part of this entire series from target selection to going for the human clinical trial. So so like from target selection, when I'm saying it deals with a lot of proteomics, and then like one of our guest lecture which was scheduled just 2 minutes back, he has also told a lot about finding the surface attribute and finding the surface property of a protein. So mostly the protein targets or enzyme targets can be tackled down by using the structural biology, and followed by the lead discovery, which has a lot of high throughput screening. And to make it in silico, we have the virtual screening. Then you have right now so much of kits which can tell you that how synthesizable one chemical moiety can be. So that means The chemical synthesis can be AI backed, and it can tell you that what is the feasibility of a synthesis, what is the potential stability of a reactant or chemical agent which you are creating out of the synthesis, and followed by you can do a lot of create a lot of models on which you can try your drug molecule. If even before going to the synthesis, you can do a lot of in silico mining, some sort of it can actually take back and it can replicate the idea of doing a series of in vitro and in vivo testing. Okay, so this is just a typo mistake. This is in vitro and this is in vivo, and then you have there are so many of clinical trialopedia data which are available, which can give you a meta-analysis that how good or bad this particular drug can be into the final state. So when I'm saying that right now data is abundant, the reason being is that we'll try to take this entire CAD perspective into three different domains. First one is the very base, that is the ligand structure or chemical structure, which are essentially the micromolecule or the small molecule. And when I'm saying that we need to deal with a lot of this thing chemical structure, it is not only the 3D coordinate point, it's also the information about the force field, its trajectory, and sometimes you can take the RMSD, that how they are superimposing if you are using something like a pharmacophore, or you can also have multiple orientation map out of this entire thing. So if you have a handset of molecule like 10 or 20 of them for a particular target, then I can find a essentially a steric conformation which gives you some interaction, and we say this is pharmacophore. And in addition to that, we also talks a lot about structure and so structural biology. It has to deal a lot of coordinate information, all these like adjusting this structure through some in silico statistical paradigm, like which has been discussed in last session. It's like predicting the R value and G value of a particular structure. And then you have in protein, you can also represent the protein as sequences, interaction map, and furthermore metadata and pathway models. That means which particular pathway this particular protein is a responsible candidate into. And also apart from this, you have a tons and tons of data which are coming from the genomics, especially say your RNA-seq data, methyl-seq, ChIP-seq, and then taking this data, we can do all this association study, and then we are saying this is genome-wide association study, which try to take the SNP, thousands of SNP at one cohort, and then we can always correlate this cohort or the set of SNP as a prognostic marker. So when I'm saying genomic data, it has to deal with stream, metadata, SNP, and biomarker as well as pathway model. Now we'll simply try to touch every point once to see that how the holistic way of mining the data has given into new project, new insight, and new inference into the entire system. So now the question arises: if you have you all are right now, we are having handheld devices which can take care of your blood glucose level to other heart condition, like you can have a handy gadgets which is like your smartwatch, which can tell us a lot about your activity, where physically you are right now there, which GPS tag you are using, your age, sleep indexes, stress, and health. And similarly, we have a lot of data which are coming and pouring into every day. It's genetic, epigenetic, microbiome, metabolomics, and proteomics. Now, what exactly we can do with this data? I mean, first we'll see what are the challenges, then we'll try to see that how combining different data, the inference system is getting more and more accurate, as well as it can be backed by a series of artificial intelligence based big data analysis. So when I'm saying big data analysis, it's basically a series of algorithms which has been designed to inherently deal with some of the data which goes much beyond a few terabytes. Like we'll see some of the example and how to do it. So, I mean, you all will agree with me with this term that drug development is a time-consuming and complex journey. Otherwise, there wouldn't have been much more the last 30 or 35 years, people are using the in silico approach to make this process much more faster. And we have seen the result in last 2 or 3 years during the COVID. The number of manuscript or the article published and the number of small molecules which has been repurposed from the known database as a candidate molecule for the this particular COVID-19 has also given and paved a way that in silico analysis is one of the most effective and fastest way to go ahead. And since there is a high uncertainty level associated with this CAT, so we have various high throughput technology which try to currently use this multiple omics data. So you have genomics, proteomics, and then your clinical trial cohort. You can combine it together to make your inference system much more robust. So I was going through one of the recent review which says about that the use of raloxifene, which is being used as an invasive breast cancer in 2007, and similarly depoxetin also in 2012, and the use of aspirin. This is actually most recent one in 2015 only. It has been seen through the EHR, that is electronic health record. So every patient has a health record, and if you have the central cloud where all this EHR can be stored, people have seen that the use of aspirin was initially for analgesic, but then by integrating all the information which was available in EHR, it has been recently found that aspirin can be also a potential ligand which can be used as a treat to treat the colorectal cancer. And in 2015 only, this US Preventive Task Force has recommended the use of aspirin to prevent colorectal cancer. So before that, nobody was have used it, but then the EHR the changes of this patient, and their I mean, this is accidentally this has been happening, that aspirin has been given to few of the patients who are into the primitive or the first initial stage of colorectal cancer, and they have found that although it has been given as analgesic, but then it has given some you know effect of eliminating the root cause of colorectal cancer. So what I'm saying that it's not directly from structural biology, but with only a handful of data mining, these three success story has been recently given in last 10 years or so. Now, I can just summarize that the big data driven drug discovery has been increasingly popular strategy within all the pharmaceutical company. The reason is there are two main reasons for this. That is the availability of processor in a much more affordable way, and also you can say the availability of the hard disks or the hardware where the storage devices are pretty cheap now. So if you do remember the strategy when the Human Genome Project came into the picture, the initial idea was to spend $10 per base pair, and we have around 3 billion base pairs in my DNA, or in that way anyone's DNA. So the target was to start with it's like almost like $30 billion project, Manhattan Project, launched by the NIH, and then finally it took almost like initial the idea was to complete it in 10 years, but then it got completed before that. But then from a project of size 3 billion or 30 billions to and 10 years, right now if you go and have your genome sequenced, it will take hardly 6 to 8 hours depending on the type of system we are having. So within 6 hours you can get your entire genome sequenced, and within a 64 gigabyte pen drive they will give you, and it's cost around 54,000. It's not even 55, it's less than that, 54,000 in 2 months back. So from 30 billion to 54,000, and from 10 years time to merely 6 hours, this has somehow has one benefit, and in one way it's benefit, and another way it's also it's challenging. It's beneficial because right now everybody can think of a personalized medicine strategy. That means I need to take only that particular molecule for which has the maximum propensity to have some good effect into me. So things are getting more and more personalized and tailor-made. similarly, there is a problem associated with this entire strategy is that once you are creating 64 GB of data for every individual, so at end of the day, you are actually go on, putting into few thousand terabyte of data every day into this scenario. So I was going through one review, and it's like as a human race, we are actually right now adding 2.5 40 trillion bytes of data every day, and around 60% of the entire data set which is being added every day into this space, data space, or the cloud space, you can say 60% are from the biological domain. So that means they are mainly from next generation sequencing, RNA-seq, and all this clinical trial and cohort study analysis data. So the idea is that if you have so much of data, how to play with this or how to deal with this. There are several such challenges, and we have we know the very simple concept of when you go through. I think almost all the fellow audience which are right now attending this workshop session, they know that what is the challenges associated with this assembly process. So the idea is like any sequencing machine, if it is pretty accurate, then it has a series of, although you have right now the Oxford nanopore which can go up to 1 million and more than that, but then most of the second generation sequencing like Roche and Illumina, they try to confine the length of the string at which they can read is around 2 to 20,000 length. So you just imagine that if you have a, like if I can give an example, like something like you are having a book which has some story going on into the entire book, and this book is your book of your heredity, or this book is nothing but your book of your DNA, and the number of characters written in the book is actually 3 billion, but with whatever intelligence and instrument right now you are having, you cannot go through the entire book of 3 billion character. So the idea is that instead of start reading the book, because you cannot do at a time, so the idea is that you tore down different pages and different paragraph of the book, and you give it to a series of people, like we have right now a group of size 35 or 36 I am saying. So just imagine that all the 35 or 36 person will be given 1 pages each or 10 pages each, and everybody will go through that part of the story, and then finally somebody will call all of them, listen to their story, and try to tag it together to develop the entire story. So that is how the assembly is going on. So that means we have so many of short sequences, and then we try to combine the short sequence to come up with a bigger sequence, and then the bigger sequence happens to be substring, and then you add on all those substring to come up with a further bigger sequence, and you repeatedly do it multiple times again and again till the time the entire genome has been reconstructed. Okay, but then when you are telling that each of the pattern which you are right now dealing is of length 200 or 2000, and then you are having around 10 million such sequences. So to check that which two sequence out of this 10 millions has a prefix and suffix matching, because the story need to be also, when I was saying that I am giving one story to two people, each reading half of the story, so I don't know which part of the story comes first and which followed by. So the strategy is that when you try to tore down the book or tore down the pages, you do in such a way that the first person will go for few pages and the second person will go for few pages, but there should be a common element, that is some overlapping region, which makes this like, just for example, if somebody will say that there's a big sequence, and out of the sequence I will write something like Hari is going to market, and there is another string which is like market to buy a bicycle. So then I can combine that Hari is going to market to buy a bicycle, because I know there is a overlapping region, that is the market. So this is how we try to combine all this substring to come up with the complete string, and this process try to reiterate it again and again till the time you created the entire range of substring, and then finally the draft genome. And to help this, we have so much of graph theory. That means we can make a Eulerian graph which can simulate something like your traveling salesman problem. So from every node, if you just imagine that every node is potentially one of the substring, if I have a way to go from one node to another node by touching every node in a unique path, so that means I can combine all these substring to come up with the entire string. So these are some of the challenges which is more and more vigorous when you are having data size going into a few petabytes or more than that. Now we have seen that genomic and this entire the concept of biomarker has recently acting as a big development factor which gives the potential to predict the outcome of this disorder and any physical function along with its symptoms. And since the biomarker based cancer development has been demonstrated higher yield, that means it's actually the yield has been somehow been manifested twice a few years back. And this is just because of, like, instead of targeting one molecule or one target at a time through a series of molecules, the idea is that can I have all the information all together and then train my model or train my system in such a way that if I like a few years back we used to say this particular X ligand is somehow can be targeted to the particular protein that is a target, a potential target. But then the question arises, and in fact, if you have published anything paper, like the reviewer used to ask that, what if the same molecule is also targeting other protein into the body? Now the idea is that if I tell that it's not only the ligand which is matching with the target protein, rather if I can tell you that what are all the targets which is possible inside the body, almost all the pathway where the ligand can go on match. Now, in that way, you can actually know that overall implication of this ligand, and you know that even if my ligand is targeting this particular protein, but it's also targeting a handful of other protein which can create some problem. And in fact, that has given some very typical problem like your thalidomide poisoning, if you know that seal limb effect. Thalidomide has been given some other purpose. It was working fine. It's not that thalidomide was a bad drug, but then after a few years, it has been found that it has some teratogenicity, and teratogenicity is because that it's actually hampering some other protein, and which is actually part of the one of the essential pathway which is required to to produce the limbs within the embryo. So if you have a model where you can try one ligand, and the ligand will not only talk to only one protein, rather what are the different propensity that with which it can act into almost all the protein. And if I know what are the protein it's getting effective, and if I know a entire pathway and I can simulate the pathway, then I know that if I am blocking one protein into a particular pathway, then overall implication of this blockade can be my potential ADMET screening kind of thing. So this type of model has already been built recently, and they are trying to mimic the process of your animal or rodent model. And although most of the models which have been developed are specific to a farm or specific to a disease, but then it is giving immense promising result, which had somehow given an idea that right now people are thinking that instead of animal model, everything can be automatized, and this the reason is also the presence of AI, because right now if you have the data sufficient amount of, and then any type of pattern which is somehow buried inside the data can be mined into, and one of the very challenging problem of finding the features is also right now being annihilated by using something like your convolutional layer. And when you are using something like your DNN, deep neural network, which eliminates the process of selecting the feature, the process of training is much more faster and effective. So genomics has been a forerunner in biomarker, and it's not because that fact that gene expression profiling is technically easy, but then it tried to combine almost all the markers. So just imagine that if you have the data about the next generation sequencing data about 10 different person who all are having a particular type of cancer, then all those thousands of SNP. So initially it was SNP to disease, one to one mapping or many to one mapping. But right now it's many to many. Multiple SNP can be taken together as a prognostic biomarker to tell that whether this set of gene or set of SNP are signature in a particular disease. So one of the example is this prognostic diagnosis using 70 genes in breast tumor. it has given an idea that that if you are taking one data at a time, it's not that holistic. But if you take a bunch of data, which was not possible, I mean, idea was there beforehand, but then when you are dealing with a data size of, I told you, it's like 48 plus gigabyte per single individual, and if you have around 10 of them, so churning out all this 480 GB data into a sizable amount of time and putting algorithm and then mining it was not possible beforehand, before few years. But right now, since the processor and the devices is allowing it try to create a hell lot of difference within the entire system. So I'll just chalk out few of this recent study which has been started and launched by different government. And if you have some time, you can please go through some of the very interesting database like TCGA, the Cancer Genome Atlas. It has a very beautiful ontology driven and very systematically arranged data about all these DNA-seq, RNA-seq, and methylation array. And recently also looking into the success story of this thing, the government of India has started its new initiative, the last line, which is also known as Genome India project, where we try to, since I'm also being a part of this project, so we know this is actually 20,000 different individuals across the India are being sequenced. So going through every caste, creed, and geographical location, starting from Andaman Nicobar to very far northeast, and then from Gujarat to your southwest, and from the deep south, every part of this India and population, they are actually, they have been taken some subpopulation, and this subpopulation are getting sequenced with their consent. And this is going to create a human genome draft of India, as well as we will also try to use it for clinical metadata. That means going towards the next step, that is creating a series of personalized medicine and personalized approach for treatment. Now, the thing which is much more fascinating this day is the genome-wide association studies. Just now I told you that, like, instead of saying that it's only a single nucleotide, the SNP for a particular disease, if you know what are the total number of SNP or what are the total number of SNP in addition to the other trait, and if we know, and if we are finding similar number of SNP into some other diseases, so we can effectively use the similar drug which was in disease X now into disease Y, because I know that this number of SNP are somehow common in both the diseases. So in one way, one of the GWS not only help you to make the things much more scientifically you can say foundation is correct, but rather also it will help you to try out new targets. So in 2014, one study conducted by Okada, we try to find out 101 SNP which is associated with the rheumatic arthritis. So, and in fact, you'll find currently the 18 out of 27 approved drug for rheumatoid arthritis, they actually use the same data suggested by this particular study. So what I'm saying that when we are trying to take a set of SNP which are associated into every person who is having a particular disease X, then I know that this SNP are obviously the signature for this particular series of person and for particular disease X. So all those targets which are related to this SNP or the set of SNP can be taken as one of the potential target for this particular disease. Now these are some of this similar database. I just try to find out. It's from which are deliberately telling about which GWAS study, and then we have also one, this thing was from mouse, but then it's being used as a this is actually being used to check that what are the overall implication of one particular disease when you are using this mouse as a rodent model. So what are the overall changes which this rodent model can effectively get from this particular drug can be easily done right now by using MGI data. And in addition to that, we have a lot of pathway information. This pathway is not only about the protein, but this pathway tells a lot about, like, the information from protein to genome to protein, and then from protein to what are the different targets, and from target to drug, and from drug to adverse reaction. So there is a hierarchical level of series of data which is associated there, and then we have recently launched the first database of this to the Genome India project. This will be very soon released by IBDC, Indian Biological Data Consortium, and this has the entry about 29,000 diseases, and it has more than 3 million entry, and every disease, which particular drug it is responsible, and also which particular genome this disease is being cropped up. So all these information are there, and we have also seen that few of the drugs which are not yet targeted, there are some of the very like rare drug where you don't have funding to study those things. But if I know that that particular rare disease and one of the disease which is very well studied, if they are sharing a set of SNP through my, and I can predict this from my GWAS, then I can use the drug which are into the well-known disease as a potential ligand molecule or lead molecule into my study, which is for this particular rare disease. So all this rare disease information are right now being, you can say, taken into as a repurposing of the drug which is known to us till now, which are from the existing study. And to do this pathway analysis, we have a series of data. Most of the things are, I think, it's already known to you: ChemBank, Zinc, PubChem, and all this thing. But here, if I'll just highlight you, this is the third and the fourth line, small molecule pathway database. It's if you have some time, please go through this. It has given a very beautiful way of arranging the metabolomics pathway and human metabolomes database. They are somehow, it tells a lot about the effect which I just now told, that if you are using a chemical molecule on a particular target, what are the overall implication this target can into the entire body. So all those ADMAT toxicity screening, so that means a toxicity which can happen to someone after 1 or 2 years of taking this drug can be also studied by using this, because if you have this pathway, and if I can block a particular protein into the entire pathway, I can use some ODE, that is ordinary differential equation, to see that what are the different protein which are associated as a precursor or follow molecule from this particular protein can be also disarranged or arranged accordingly. So that means if I am blocking X, what are the overall implication of X into my entire pathway can be easily studied with some of the time series engine the software is the database is providing. Now, use of EHR, I have tell you one example, that the propoxif patient, which we are more into myocardial infarction, it has been identified that the safety of a particular clinical trial drug, that is silostazole, has been found out just by EHR, even before the clinical trial has been done. And this is only possible because once you start taking this drug, then during the clinical trial itself, before comprehending the entire analysis, the electronic health record, they suggested there is a pattern emerging that all those person who are taking this silostazole is having the similar effect in for CHF, and then in fact the same report has been also been verified through clinical trial. So there are few known example which has suggested that if you use only the EHR data, also EHR data gives a lot of pattern that particular taking one drug. So what is beyond clinical trial? Clinical trial is always confined to a certain population where the number of individuals are very restricted. But if you take EHR, it tells you across the domain, across the geography, how one particular drug looks like, and then looks like in the sense symptom wise how it looks like, and what are the overall implication of the drug into the body. And there are a very well-known EHR database is your PCORNet, and then you have also FDA Orange Book, and in addition to that you have Mimic. Mimic is from MIT, and then they have recently given all the EHR, in fact a handful of EHR free of cost. So you can try your hand by using, and they have also a series of NLP tools available there where you can You can screen down the literature which are related to one particular disease, and then literature in the sense all this EHR data, and then you can see that whether the same problem is existing into all the patients who are taking a particular drug. And if it is there, it's more like a clinical trial, but it's much more robust, because clinical trial is always a confined study, and whereas the EHR tells about the open scenario. So people has right now also associated this GWAS with the EHR. So GWAS tells about all those SNP which are associated with a particular disease, and then EHR tells about what are all the symptoms which are associated with the disease. So if we just add up all the symptom as well as the set of SNP of this particular disease X, let it be. Now it's actually a better way of saying that phenotype to so it's a phenotype wide association study. So you try to take the GWAS. GWAS will tell you the series of SNP, and all these series of SNP are somehow related to a certain effect or symptom, or you can say secondary symptom arise during the treatment. Now if you use the symptom along with this SNP, now this gives you a PWAS, or that is phenotype wide association study. And there are some very interesting work which is also going on in Indian perspective with the help of Sanofi and Indigene into this. So these are just of the protein and chemical genetic interaction, which is one of the very good one is your DIP as well as BioGrid. BioGrid tells a lot about protein, genetic, and chemical interaction. Something which I told you that you can find from genetic what are the different protein is coming, and from protein which chemicals are associated, what are the interaction, and what are the association study. You can just go through this BioGrid. Now this is a database of bioactive molecule, which I think most of us have already gone through. So by combining all together, we have also seen that since you have all those data with you, so you have chemical data, you have GWAS data, you have all this biomarker, you have PWAS data or phenotype wide association study. Now what to do with this? So you can combine it together to make your QSR much more effective. So initially, before few years, the QSR is all about finding a fitment model, a mathematical model, but where the parameter of this model will be nothing but the property or the features of a particular ligand or the small molecule, and then the half-life period or the activity will be as the target. But whereas recently there's this deep neural network which can gives you the idea of taking thousands of nodes at a time and into thousands of layers, which has given this idea that you can take all those study as a separate feature, and you can create a hefty network of DNA, and then you can do a lot of QSR analysis which are similar to the initial one, but these are much more accurate, in the sense that it's not only taking one single approach, it's rather taking a holistic approach. So these are few of this study from our group, depending on the QSR. These are mostly targeting the blood-brain effect, mutagenicity, toxicity prediction, and by using the machine learning. Now, since QSR has been right now taken into one step ahead, and we are saying this is actually not QSAR, it's actually QSPR, quantitative structure property relationship. So instead of taking the activity as a target, we take this toxicity or distribution or metabolism as one of the targets. So if I know that a particular drug is having this 10 different feature or this 10 different property, then there is a most likely value that this drug can get metabolized faster. So if we'll have the metabolic, since the data is already right now there, because this thing is a part of also BioGrid. I told you one of the databases in BioGrid. So BioGrid, from the BioGrid you can find that which particular molecule get the distribution, metabolism, and what is the rate of extinction and toxicity. You can get all those data, so you can create a model which not only take the features of this molecule, but rather it's also take the ADMET property, so that we can always predict that given a new drug or given a new drug candidate, lead candidate, this particular DNA model can make this prediction of the ADMT parameter much more faster. And you have seen this thing in last 10 years. ADMT prediction and validation are right now much more faster and much more accurate. In last 3 years, the COVID has given a very good example, like within the duration of the vaccine designing, as well as lunging few of the drug like ivermectin and other drug, which are actually nothing but repurposing the existing drug. We have seen that the ADMT parameter has been satisfactorily interpreted by in silico model, and till now there is a very good consensus between the in vitro model and in silico model. So these are few of the database which tells about the toxicity, and I'll just share this thing with you so you can just browse through. And this is the data which gives you an idea that you can build your own own model. So these are few of the already known model, and here you have the data about every drug, its associated toxicity, and so you can take this as a QSPR scenario. So you can use your own model to create the toxicity prediction network, like you're using your ANN or any ML algorithm like your random forest or decision tree. So when you try to train this thing, I told you that since the DNN gives the idea that you can not only train the small molecule, you can also train the different property of protein and associated biomarkers. So to train this protein, you need to somehow quantify the protein into a series of features. So these are actually a series of database or tools which tells what are the different features of a protein molecule, so that every protein molecule can be essentially taken as a series of vector. And then if you have the protein, you have the corresponding genome, and you have the corresponding drug, you can take it together with the GWAS study to make your model much more accurate. So I'll just try to summarize everything within 2 minutes. So we have seen that the accumulation of drug knowledge, multi-omics data, and then clinical data, which comprises of cross-domain big data, it facilitated the systematic drug discovery in a much more faster way. The increasing informatics infrastructure has somehow enabled us to do this analysis, because one of the example I gave you, Genome India, which is actually Government of India project, we are supposed to come up with 4 petabyte of information. So 4 petabyte is actually a humongous number of amount of data, which if you want to study it through the traditional algorithm, like even if you have information about every 20,000 population across the India, but if I'll tell you that how similar is one fellow from Nagaland with one fellow from this Allahabad or Uttar Pradesh, I cannot do it simply within a logical time, because we have to deal with all this all these 4 petabyte data. But right now, since we can distribute our computing into across the different node, and we have some already inbuilt tools where you do, as a bioinformatist, I don't have to need to know each and every details about this thing. If I know how to write a few lines of Python, and if I know how to deal with Spark, then there are so much of tools which helps us to do the analysis, and there are already known program or function for this. You can directly call the function, and it can do the remaining things such as the genomic, proteomics, and GWAS analysis on big data. We have also seen that numerous well-defined and high-quality clinical phenotype information are greatly somehow facilitating the construction of a model, and which I told you that the model are much more accurate. It can give some new discovery or new screening molecule which can have low toxicity, and in the same extent it can also help you to bypass the existing rodents. And to summarize it, we have also seen that rapidly developing machine learning technique have somehow drastically accelerated the entire paradigm of drug discovery, and it is actually promoting further performance of both de novo as well as repurposing. So de novo means creating a new lead molecule, and repurposing is nothing but if you have the information about a particular drug, like the beautiful case is ivermectin. People have used this ivermectin multiple times during the COVID, but you know that ivermectin is actually for worm infection. So it's like something like your albendazole and ivermectin we used to take for one, but people have used the ivermectin for COVID, because this is I know one of my friend who is in Dr. Reddy's. They actually did some in silico study, and then they found that ivermectin is having a good binding on the virus protein of COVID-19. So and then the incorporation of direct consumer genetic testing, that means the test which everyday people are doing, they are loading in the cloud, and this information can be taken off and can be associated with the already known patient as a cohort analysis to find the GWAS. So that means if I know that I have all those 40 or 50 biomarkers into me, which has been somehow as a prognostic biomarker of a particular disease or cancer, then there is a high chance that I may find or I may develop this particular type of cancer a few years down the line. And in fact, this is actually one of the reason you have some Hollywood celebrity who got their I think I forgot the name. I think Angelina Jolie has got some surgery even if before finding the breast cancer, because the prognostic marker of breast cancer has already found out or into the direct to consumer genetic testing which she has done, and then she decided to go ahead with the surgery even before going into the paradigm of finding a cancer. So that's all. These are few of the tools and software which you can browse through. They all are freely available. You have scikit-learn, PyTorch, TensorFlow, Weka, and 9. And then these are few of the books which are also available. I'll just share this book information and this information with the organizer of the workshop, so you can, whenever you are free, these all books are pretty explanative, and they all gives with some example. And I think as a bioinformatician of this generation, you all should know at least one or two tools out of this set of tools here, because I know down the line every data is getting into the big data scenario, and once you know the way how to deal with this data, you will be the winner. So that's it, guys, for today, and thank you for all this patience and hearing. This entire slide and everything has been done at my lab. This has been done by a few of my students. So I'm only one who have gathered and collected everything and put into one slide. So I'm open for question. If you have anything, you can please go ahead. Thank you.
Participants, please ask questions, clear your queries. You can ask, or if you are not feeling comfortable, you can write it in the chat box.
Yeah, good evening. Am I audible?
Yeah, you are audible, Chetan. Please go ahead.
Yeah, good evening, sir. Yeah, it was very simply talk. Well, I had
Chetan, is it
one
the
question.
same Chetan from Pondicherry?
No. This is from Gujarat, sir. I'm from Gujarat. Okay, sir, you mentioned about the database where you were trying to the database shows us what are the interactions we at molecular level with the protein as well as the ligand. But sir, if you take some examples, like there are cascades also in many of the diseases. If you observe, there are cascades. Cascades in the sense, one classical example we can take as an hemophilic factors, where there are different proteins which are getting activated, a protease, set of protease which are getting activated and causing the subsequent cellular reactions which are responsible for the a particular disease happening or not happening there. So how does this model, what you have mentioned, help in understanding what are the different cascades which are involved in a particular disease?
Okay, so if I do understand your question, you are saying that what will you do when you have a cascade of reactions which are triggered by this particular small molecule? Yes. So when I'm saying that there is a cascade of reaction, they are essentially nothing but they are actually this pathway information is already there with you. I mean, if you know that if I will somehow block this particular protein, so the protein doesn't although the protein try to alter so what happens when your ligand attack? So when the ligand is getting attached, it actually essentially may change your active site or some region which is near to the active site or overall attribute of the protein, and due to that there is a chain of secondary metabolites or the primary metabolites or tertiary metabolites, they try to increase or decrease. Okay, that's how the things
Okay.
goes. Yes. So if you
Right.
if you
Yeah.
if you remember the when one slide I told that there is a human metabolome database, HMDB. Yes.
Yeah.
And then
Yes.
this,
Yes.
and also there is one database known as pharmacogenomics database. So if you see
Right.
this pharmaco and KEGG also, you have KEGG. So KEGG and
Yeah.
human metabolome database, they have also essentially all the information about the primary, secondary, and tertiary metabolite. So
Okay.
the idea is that here the protein is connected to another protein through some secondary metabolite. So just
Right.
imagine that if I'm targeting A is supposed to release some
Mhm.
chemical messenger, and due to release
Okay.
of this chemical messenger, B is got triggered. Okay. So if I'm
Right.
blocking A, then the
Mhm.
triggering of the B is not possible here. Yes. But
Okay.
essentially, if you see in a pathway perspective, A is connected to X. X is that particular chemical messenger, and X is connected to B. Okay. So if
Okay.
I'm blocking the A, I can know that what are the total number of blockade in X possible and how the B will be changing. So
Right.
they also gives you some ODE solver, that is differential equation solver, where you can block and you can see that how through different time scale the entire set of B is changing. So if you decrease the A, you see that there is an increase in B. So previously increase in A was decrease in B, but since you block this A, now the B can further increase. Yes. So all these things you can always study through ODE solver, and that is also there in scikit-learn. So you
Okay.
can check this.
Let's take an example of this since an ancestral pathway of clotting. You might be aware that there are two different pathways which work on for clotting, right? So there may be certain disorders where you see that multiple pathways are working. So how does this play in there? Because you may not be having the A or B or C factor involved there. You may be having altogether different factor which is involved. I'm just trying to understand.
Yeah, actually,
Does this?
what happened, like if I'll say that any chemical reaction, like you are saying A and B, two proteins are there, and there is a presence of a particular messenger. Okay. So I can convert this equation into a stoichiometric equation. So I can find what is the
Okay.
concentration of A with time. If I know the concentration of B and AB with time, yes. So the civil the Michaelis-Menten reaction, yes, if you do remember. So that is actually,
Yes.
yeah.
Yes.
So you convert into a stoichiometric equation, and once you convert into stoichiometric, you can actually differentiate it with time, and you can see the how what are the different changes in the cascade possible through the time. Okay. So when you say that dA by dt, that means the change in A with respect to time will be nothing but a particular value. Okay. And then you try to take
Okay.
this time series from, you can iterate this thing for 1,000 iteration to see that how across the time the secondary messenger or this particular
Well,
B
that means it's
reaction.
essentially in correlation of the data, right? That's what it's.
It's a correlation of data through time series. It's a temporal correlation of data.
Okay.
Yeah.
Okay. Thank you. Thank you, sir. Thank you.
Yeah. Anything else? Any question?
Participants, any more questions? So if there are no more questions, then should we end the session? This is the final call. Please, if you have any questions, ask them right now.
Am I audible? Hello.
Yes, yes.
Uh, okay. I have a question, sir. Um, the recent science has been moving towards the direction of a more holistic approach, such as in systems biology, as you mentioned that you can use ODs to actually mimic the whole pathway or the network of the pathways. So, still, how do you see the importance of a single molecule interaction, such as protein and ligand interactions? And will this holistic approach will be able to match the accuracy of these type of interactions? Because we know that through the experimental validations, such as the experimental PDV structures of protein and ligand complexes, they are very much accurate. So how do you see this change, I would say, that and what would be the accuracy of the predictions using the holistic approaches?
Thank you so much. It's a really nice question. So the if I do comprehend what you are asking, that is this particular holistic approach is going to, take away this existing approaches, and whether it will be more accurate than the existing approach or not. Yes, that's what your question is.
Yeah,
Okay.
yeah,
So,
somewhat exactly
so
what you said.
it's like look, we are not going to throw away whatever we are right now doing. In fact, we are saying that whatever right now we are doing, we are taking a protein X and putting some 1 million different type of molecule as a virtual screening, and then we found some of the potential lead candidate, and then we try to make the docking one by one. That's what we are right now doing. Yes, but what I'm saying that apart from this, with all those information, if these things would have been much more simpler like this, then we could have already drug for every target, but that doesn't happen because this particular ligand may have a very good interaction with a particular protein of X, but then when you try to take it into the next step, that means synthesizing, going through ADMET, and then going for a market analysis, doing into the rodent test, it badly fails. The reason being that we have started with a simple assumption, with a simple hypothesis, and finally we found that the hypothesis was not correct. Now there is a slight change. You do whatever right now we are doing, the traditional approach will be there. But in addition to that, if you take all those inference model, now then your propensity of finding a lead out of this will be much more faster. So it's like the existing traditional approach plus a AI model, which also give a consensus that whatever right now you are doing is good enough. If whatever you are right now doing is having a very good docking score or binding score, but then up down the line you find that the model is performing very bad, then we know that although this has a very good binding score in silico, but this may badly fail in later part. So the idea is that if you want to make this thing much more faster and cost effective, so your hypothesis should be much more robust. So this holistic approach is one way to make your hypothesis much more robust. I hope I cleared it out.
Yes, a nice answer. Thank you.
So participants, are there any more questions?
I think, ma'am, they all are ready to interact with you because I think you have interaction after this. Yes.
Okay, okay.
So everybody is ready to interact with you.
Okay. So
Thank you so much. Thank you so much, ma'am, and thank
thank you,
you.
sir. On behalf of all the people on the organizing committee and participants, I thank you for this wonderful talk and taking out time from your busy schedule. As
It
a
was my pleasure, ma'am. It was my pleasure. Thank you so much, you and Dr. Sintu. Thank you.
okay. So as a token of appreciation, we present you this virtual memento. And it was very informative talk. Thank you so much. So,
Thank you, ma'am. My virtual thank you to you.
participants will ask for some more time during the offline session from you. Then surely we will be asking you to give some time. So thank you so much. So participants, we end today's session at this point. Tomorrow we will be meeting again at 7 o'clock sharp. Tomorrow we will be having 2 lectures. One from Dr. Muhammad Ataul Islam from Big Sport, Bangalore, who will be delivering talk on pharmacoinformatics, and Dr. Ruturaj Purud from CSIR IHBP, who will be discussing computational interventions for target and lead identification. Both the speakers are well versed in the area and have lot of experience, so I hope you will enjoy their lecture. Please do join 5 minutes before the scheduled time. And tomorrow we will be sending you MCQs also that you are supposed to answer and submit. So tomorrow will be the last day for the online session, and then we will be meeting on the campus on Monday for the offline sessions. Do mark your attendance in the Google Sheet provided to you. So thank you everyone for patience listening. Thank you so much. We will meet again tomorrow. Good night.